Hey folks, we're going to go through Animals 1 specimens with you today. So starting with some sponges, I just wanted to say hi real quick before we get started. So I'm going to go ahead and share my screen for the sponge specimen that we're going to be looking at here. Okay, so we're starting with phylum Porifera. And this is a parazoan, which means that it has no true tissue types. And you're probably familiar with this. So this is just squish, 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 like a regular commercial sponge, right? Soft, has some nice spongy texture feel to it, all right? And so when we're talking about sponges, we like to kind of think about sponges having like that really nice opening at the top, the osculum. But this one, it doesn't have a really nice, perfect, just singular hole at the top. So this one's a little bit different than our model organism for a sponge, but that's okay. And I kind of wanted to just give you the feel for this one in comparison to the other one. So this is Dimnospongiae. Okay, so that's one of the classes of sponges. And I also have a little piece of the calcareous sponges. So if we look at this sponge, and I'm just going to try and zoom in here a little bit, you can see that it's really not very visually interesting, let's be honest here. But it's much harder than this commercial sponge. That's because as a dried sample, it's mostly just calcium carbonate, okay? So that's going to be that type of sponge. And then we also have this third type here. So, ooh, this contrast here is horrible. Give me a second. Okay. And so this sponge is really delicate, so I don't normally take it out of the plastic, but because we're on video and it's really hard for you to see otherwise, I'm going to take it out. So, and this is an example of our glass sponge, okay? So this is that third class, Hexanelladinae. And this glass sponge is really, really delicate. And you can see that actually, honestly, from my end, let me just show you this piece. It's almost like, feels like fiberglass down here, okay? And that's because this shell here that's left over from the sponge itself is made out of silica, right? The same thing that glass is made out of. That's why we're calling these a glass sponge. All right, so those are our three classes of sponges. And when we're thinking about the classes of sponges, they're all still going to have the sponge body type. So I was thinking maybe we'll look at a sponge underneath the microscope. Okay. And so let's get focused in here. So we're looking at a sponge underneath 4X, and I'm going to start at the very bottom. Let's get zoomed in. Okay, so sponges are kind of big. So when we're looking at them underneath a microscope, we can only look at a little piece at a time. So let's start at the very bottom. Okay, so here's the bottom. This is what would be connected to the substrate down there at the bottom, right? And as we move up, again, nothing really super crazy to look at. It kind of looks like popcorn fluff to me. And that makes sense because the sponge doesn't have true tissues. So sponges are made up of a bunch of different cells that are working collaboratively, but they don't have a true tissue layer. And so what we're seeing is you can kind of see, look on the left and look on the right, you can kind of see how it's zigzagging in and out. Do you see that? So that's going to be where you're seeing those pores that the sponge has. And so this is a longitudinal section. So what we did was we took a nice thin cutting of a sponge. And so what we're looking at here is on either side, you're looking at the fibers of the sponge, right? The spongin probably. And then you're seeing the pores, that's those kind of indents where the zigzag is going. And then in the middle, there's this big gaping hole. Okay, and that's going to be the spongocyl, or the main cavity of the sponge. So let's keep moving up. Still got lots of pores, got that big cavity, the spongicyl. Keep moving up. Okay, and then at the very top, it got curved, like that's just part of this microscope slide. That's not how it normally looks. But at the very top, you can see it's open, and that's going to be your osculum, or that top opening where the water exits the sponge. So I'm going to go back through, and I want you to take one more really good look at how this sponge looks, how the fibers look, and how can you tell that this is a sponge if this was, say, a lab practical. Like I said, to me, it looks like popcorn fluff, and it's got those zigzags on either side, but otherwise it's pretty disinteresting, all right? And that's just my opinion. That's how I remember it. But I want you to figure out, how do you remember that this is what the sponge looks like when we're looking at a longitudinal section underneath the microscope? So let's go down all the way to the bottom. I want you to have one last really good look here. Okay, so sponges. All right, next we're going to take a look at the next grouping here. So we're going to look at cnidarians and ctenophorans, right? And so these are organisms that are diploblastic, meaning that they have two cell layers. And these specimen jars are like from the 70s. So I'm not going to probably open this because it would be foul, but I'm going to tip it over for you, and hopefully you can see. So this is a man of war jelly, and you can see on the top is kind of this balloon type thing. And then on the bottom here, you can see that there's lots of little things hanging down from it. And those are actually going to be the tentacles. All right, so that's one specimen we have. This one here is a moon jelly, and you can see kind of the cap on the top, and it has very short tentacles. It's actually just like this last little inch here. So that's a little bit different morphologically, but same basic. And I want you to think, while we're looking at these are examples of cnidarians. And are we looking at the polyp form or the medusa form? So I'm going to show you one more, and then I'll tell you. Okay, so we'll look at this one. Actually, that one's pretty difficult to see, so let me choose a different example. Ooh, how about this one? How about this weird one in the Nescafe jar here? So if I tip it back, oh, it's kind of going with it, but you can see the top. What you're seeing is the top, or the bell part of it, okay? And then if I tip it back, what you would see is some tentacles down here at the bottom. So each of these are different examples or specimen of the medusa form, right? So in the cnidarian life cycle, one of the forms is the medusa form, and that's the one that has, let me grab some chalk here. So the medusa form, right, we have our little cap, and then we have our tentacles. Okay, so that's the medusa form, and that's what we're looking at in these specimens. So we've got this bell-shaped part, and then we have the tentacles hanging down, okay? So there's another form in the cnidarian life cycle, and that's going to be our hydra form, or excuse me, it's going to be our polyp form. And one of the ones that we can look at for that is a hydra. Let me find the microscope slide for that. Okay, sorry about that, I found it. So we're going to do the same thing that we did last time. So we're going to start at the very bottom, okay? And so here we're looking at our hydra. And a hydra is a type of cnidarian, and it's generally smaller, as you can imagine, because we're looking at it underneath a microscope, right? So here's the base. That's the part that would connect to the substrate. And as we're moving up, this is really long and squiggly, but eventually we're going to get all the way up to the top. And it's got these tentacles, okay? And so with the polyp form, it's the upside down version of the medusa form. So instead it has a long stalk and then the tentacles face upwards, okay? So if we went back to our drawing, thanks assistant. We're gonna have that long piece and then the tentacles, okay, up top. So they're just inverted versions of each other. So tentacle, tentacle. I'm just going to call this the body of it. Just different. I mean, right, this is like the bell shape and this is like kind of a stalk shape, but it's the body generally of the organism. And what we'll find is that both of these have an incomplete digestive system. So the mouth for a medusa form is going to be right here. And then this is going to be the incomplete digestive system that lines kind of here. And then in this version, let's just kind of do this. This one, their mouth is going to be down here, right? And then digestive system, incomplete digestive system is right in here. So these tentacles are going to feed the mouth, any particles or anything that it catches. And in this case, these tentacles can feed stuff into the mouth here. And what we find is that in both of these forms, when they ingest something, it goes here into the gastrovascular cavity, and it's digested, the nutrients are absorbed, and then guess where it goes? Well, it's an incomplete digestive system, so the mouth and the anus are the same thing. And so when it's done digesting something, it just excretes all of the waste back out through the mouth anus, okay? So these are kind of funky. This is really different than our system, but these are highly effective organisms in the environment. And so kind of just cool to think about a different kind of system here. So the medusa and the polyp form, and a really good example of the polyp form is our model organism over here, the hydra. So just want us to take one more good look at our hydra. So tentacles, making sure that we're taking notes about how are we going to recognize this on the lab practical. I think for me, at least, the microscope slides can be the trickiest to identify, but it's pretty straightforward here. We've got the tentacles on top, and then we have a nice long form down here to the bottom, and that whole length of that organism is going to have the gastrovascular cavity going down there. Sorry, I got lost in my microscope slide. It's backwards. Ah! Okay, and then we get down to the bottom, and that's the base that's going to connect to like a rock or a substrate or something. Another really great microscope slide that I have for this is called obelia. And obelia is another great example of a hydra type form. And this type form, which I think is really cool, is it has these polyps. So again, let's go down here at the bottom. This is the bottom, but it's not really. This isn't what would connect to the bottom of like the substrate. This would be more like they snapped off a stalk. Okay, so there's the stalk, and then we come up here in this first form, that's a little polyp. It's not very mature, so you can't see its tentacles, but let's keep going up, okay? Oh, look, and so that shape there that's kind of curving over and pointing downwards, that's gonna be part of this obelia, which is it's kind of like a stalk with different colonies on it, okay? And in this case, these polyp colonies can form, see how the polyp is kind of right down there at the tip of the stalk, or I guess the branch. At any point that could break off and then become the medusa form, okay? So it would just stay upside down like it is, right? And then it would float through the water column. So let's take a look and see if we can see any others on this particular stalk. So here we have one up here. You can actually see two of them here. And so in this form, when they're connected to all of those branches in the stalk, that's going to be a polyp form. It's sessile, right? It's non-mobile, okay? But if that tip popped off, it could invert itself so that the tentacles are down. It would be a medusa form and it could float through the water column. So these are just really, I like it. It's confusing because it looks like a plant, but it's not a plant. Remember, it's an animal. So it's a cnidarian, and this is the polyp form, but these little polyps can separate from this colony and become lots of little medusas. Maybe one of my favorite microscope slides in the 1802 lab. Okay. So the last thing I wanted to look at with respect to cnidarians are some coral samples. And I don't have any live coral samples, but I just thought we'd look at some of the casings that are left behind when corals die. And so corals are colonies of lots and lots and lots of tiny little cnidarians, specifically polyps, and each of the polyps creates a little calcium carbonate shell around it. And since they're living in colonies, as they create these shells, the shells combine to form these coral structures. So some of the coral structures kind of remind me of some of the fungi we looked at last week, right? The coral fungi. So they can create these like long kind of stocky forms. And then we also have some kind of like brain-like structures. Okay, and so all the holes you're seeing here are where the little polyps would be if this were live coral. I also have this really crazy coral sample. This one is big. Okay. So, but you see all the holes? Like you see how it's speckled? And every single one of these holes would be a little polyp or a little member of this coral community. So in this particular coral, right, there would be hundreds, maybe thousands of little polyps all in here. And then they're creating a little calcium carbonate shell that they live in that protects them from the elements. And so in the hole, imagine there's like a little guy and he's got his little tentacles floating in the water column, and he catches some food and he ingests it, right? And then maybe something scary swims by and he can tuck back into this his little shell. So lots of little ones all doing that here in this colony. So those are our cnidarian samples. So we looked at some medusa forms or jellyfish. We looked at some polyp forms. We looked at hydra, which was that singular one. And we looked at obelia, which was that colonial polyp kind of tree thing. And then we looked at some corals, not live corals, but the remnants of some coral colonies. All right, so let's move on to our flatworms. So flatworms, we have now gone into our triploblastic organisms, meaning that they have three different cell layers. And so our triploblastic organisms have a ectoderm, a mesoderm, and an endoderm. And of course, the endoderm this entire time, when you're looking at the diploblastic organisms, which were our cnidarians, the endoderm is always going to be the gastroderm, or the digestive system pretty much. And then the ectoderm is always going to be the epidermis, okay? But with triploblastic organisms, the mesoderm becomes a layer that can form things like muscles and nerves and organs. And we're going to start with looking at flatworms. This is kind of an interesting one to start with because they're flat, so it's hard to think about them having this mesoderm that forms all of these things on the inside, but truly this does have organs and such in the inside. And our flatworms are acoelomates, meaning that they have little, well, not little, just none at all coelom, no cavity for organ development. So it's kind of hard to see here, but tapeworm, very long ribbon-like worm. All right. And we're going to take a look at a microscope slide of a flatworm, and this flatworm is called a planarian. So in this planarian, we can see, let's see if I can get the contrast a little bit better. All right, there we go. So you can see the green outline, and it is kind of translucent. And that's because it's a flatworm. So it really is just absolutely so flat. And flatworms, you can see what we're looking at right here is the head portion. And I know that because do you see the two spots at the very front of this organism's head? Those are actually eye spots. So, and if we looked at the tail, let's just look at the tail for contrast here. So if we looked at the tail, we can see that it does not have eye spots. So that's the tail. I know that because it does not have eye spots. And then we do have a tail here, all right? So this has the eye spots, and this is the beginning of having bilateral symmetry. So I didn't mention this with the cnidarians, but they have radial symmetry, and these have bilateral symmetry, right? And so with bilateral symmetry, what happens is that you have this concept called cephalization, and that's the idea that the head portion of the body forms. And so even with these very simplistic flatworms, the head portion is differentiated from the other parts of the body here. And the other thing that is really interesting about these flatworms is they're very simple, but they have 3 different cell layers, and that really allows for us to have good organ development. All right, and so what we're seeing here, all of that black crazy funkiness in and out through here, that's going to be the gastrodermis. So that's going to be different parts of this elongated part of the, oh, maybe it might have stopped the spotlighting because of that, maybe not. Sorry, some tech issues possibly here. So, so what we're looking at are all these black squiggly lines are kind of this, it's all of the gastrodermis, right? So it's all of the digestive system, and it goes all the way throughout the entire body of this organism. Why is that? Well, that's just because it's distributing all of the nutrients to all of the tissues in the body. And how do we do that as humans? Well, we have a circulatory system that takes the nutrients that we absorb from our food and it distributes it all throughout our body. Well, with flatworms, they don't have a formalized circulatory system. So they have to spread out the nutrients by literally just having their stomach in every single aspect and space of their body tissues. Okay, so their stomach is reaching to all of their tissues to make sure that all of the tissues get nutrients. Now, how does this organism breathe if it doesn't have a centralized circulatory system or respiratory system? So this organism is very simplistic. It also does not have a formalized respiratory system, and it's very flat. So what happens is, just as nutrients are dispersed through diffusion, so is oxygen. So oxygen just diffuses straight through the tissues, and they're able to absorb oxygen in that way. So taking a look, this is our whole one. I'm actually going to show you this on the stereoscope because I think that if you can see the whole thing at one time, it might be good. So let me just take a look real quick. So let's do a little frame shift here. Ooh, perfect. Okay, and I want, nope. Might just be too small. It's okay. It's kind of nice that you can see how small it is, like in comparison to my finger, for instance. All right. So it's pretty small, but it's not quite, it's like in that weird in-between size where it's not that great to look at under the microscope because you have to look at it one little piece at a time. But under the stereoscope or our dissecting scope, it's a little bit too small. But one thing that I can tell you is the tail is tapered. So even when I'm just looking at it from this view, I can kind of tell what I'm looking at. So there's the tapered end, that's going to be the tail. And then here's the less tapered end, that's going to be the head region. I'd also like to take a look at our planarian as a cross section. So we just looked at a whole planarian, and let's take a look at a cross section. I've overshot it there. Okay. I don't really like that sample as much. Let's take a look at this guy. All right, this is a really great sample. Okay, so here's a planarian. This is a flatworm, and we cut it into cross sections. And so what you're seeing is we're going to go to one side. So here's the little tip. That would be like his left side or something. And then let's go to the other side, the tip, that's the right side. So they're a little bit flatter on the sides than they are right there in the midsection. And one of the things that you will learn from the handout and the PowerPoints and all of that is that planarians have a feeding tube, a pharynx, and the pharynx extends into the actual body cavity. So that circular thing that you're looking at there right in the middle is actually the feeding tube or the pharynx. It's like a muscularized feeding tube. And I like to think about it kind of like an elephant trunk. Okay, so it like can pick stuff up and it can eat with it and it's very muscular. All right, but unlike an elephant trunk, the elephant trunk kind of stops at the front of the elephant face, right? This kind of goes into the body cavity. It would be like as if the elephant trunk extended down into the esophagus and then all the way into the stomach. So this feeding tube goes all the way into the stomach, basically, of this planarian. And what you'll see is that it has all of the different 3 layers that that we think of with the body, the 3 dermal layers. So the pharynx itself, that circular thing in the middle, is going to have the endoderm, which is that squiggly middle part that has the kind of the cavities that you can see in it. And then the part in the middle is the mesoderm, so that's the muscular part. And then the ectoderm is that outer layer or the outer circumference of the circle. So the planarian body itself, which is kind of this oval shape. So we were looking at it earlier, the overall thing. So here are the tips, right? So that outer surface is going to be your ectoderm. And then the inner part is the mesoderm. And then the inner part where you can see these kind of cavities dispersed throughout, and you can actually see them. You see how one is right next to the pharynx, and then it gets a little bit smaller, and that's next to that, and then it's a little bit smaller, and that's next to that as you get closer and closer to the very edge. So those are all different branches of that gastrovascular cavity that we were looking at earlier. So remember we were looking at the planarian, and I was showing you all of those black squiggly lines that go all throughout the body cavity. That is that's what we're looking at here. So the gastrovascular cavity has lots of different channels, and it goes throughout the entire body of our planarian. I want to go back to looking at the original one that I skipped over because I said I didn't want to look at it. Notice that it does not have that big circular structure in the middle of it. So remember, that was our pharynx, that was our feeding tube. And the thing about this is that it no longer has the feeding tube in this section. So this was probably like a section that was a little bit closer to the tail, a cross section closer to the tail, and the feeding tube just doesn't extend through the entire length of the body. So this particular cross section is missing that. But what you can see is, of course, the ectoderm, that outer part, the mesoderm, which is really just all the pink that you're seeing, the musculature. And then all of those different cavities or spots where it's lighter, a little bit lighter pink or even white, those are cavities literally of the digestive cavity, okay? So if it has a cavity, how can it be an acoelomate? Well, that's because the digestive cavity doesn't count when we're talking about acoelomate, coelomate, and pseudocoelomate. So these organisms, flatworms, are acoelomate, meaning they do not have a cavity within their tissues except for the gastrovascular cavity. And all organisms have that, remember? So even really our diploblastic organisms, our cnidarians had a digestive cavity, even really kind of sponges on some levels. So the digestive cavity doesn't count when we're talking about acoelomate, pseudocoelomate, or straight coelomate. All right, so now we're going to move on to our flatworms, and flatworms, or sorry, to our segmented worms. My apologies. So I pulled out 3 different samples of segmented worms. So just wanted to zoom in here. They can be kind of, I mean, you know, worms aren't everybody's favorite, so it's no big deal if this kind of grosses you out. But we have 3 different specimens here, and all of them are different versions of a segmented worm. So here we have a ragworm. And you hopefully can see that it has little segments, and it has little like little hairs that stick out on the sides here, but each of these different hairs also represents a different segment of the organism. This is terrifying. This is actually like a humongous leech, okay? I mean, that's a very large leech. I'm just giving my finger for size here. And so the leech is a segmented worm as well. And then the last one that we'll look at here, this is an earthworm. And I'll do an earthworm dissection here in a little bit, but we're just going to look at the specimens here first. And I think you can see it really clearly here. So each of the different segments of each of these different body types. So all of these are segmented worms. And segmented worms are true coelomates, meaning that they have a true coelom formation. And when we have true coelom, that space where the coelom is and there's communication between all 3 of our different tissue layers, we have pretty complex organ formation in response to that. So let's take a look at what that looks like underneath the microscope. Oh, would you mind to get back on so that we can spotlight? I think it kicked you off. Okay, it's okay. All right, so let's take a look. Here is a cross section of an earthworm. And again, this is kind of funky because we have to look at it. This is under 4x, and we have to look at it one section at a time. So let's start from the bottom. Well, first of all, you can tell that this is really a lot more complex, right? So this is, remember when we looked at the sponge and it just looked like really cavernous and like popcorn fluff? Well, now we can see that there's a really high amount of both tissue and organ development. So just right down here at the bottom, what we're looking at is a really thick layer of tissues. So on the outer layer, we have our epidermis, right, our ectoderm. Underneath that layer where you can see the fibers going in the lengthwise direction, those are our circular muscles. And then we also have this really thick layer that we're looking at. That's the longitudinal muscles, all right? So they have 2 different kinds of muscles, which is indicative of just complexity, right? That's more complex than one type of muscle. And that's what happens when you have that coelom formation. Things become more complex. So let's look at how else this organism is more complex. Okay, so we're gonna move up here. And what I'm trying to get to is this middle part, okay? So see this big hole right in the middle. It should be right in the middle of your screen. That's actually gonna be the cavity of the digestive system. So that's gonna be our gastrovascular cavity. And since this is a cross section, all of the kind of pink kind of circular thing surrounding that, that's going to be, that's the digestive lining itself. So that's our endoderm, that's our gastrovascular lining. And so the cavity is on the inside and then the lining is on the outside. All right. And then all of the space around that, which you can see, it's kind of thin here and it's a little bit bigger there, and then opens up a lot on this side. That's all going to be your formal coelom, right? The actual coelom or the cavity that's formed in coelomates. All right. And so when we have that formal cavity and it's called a coelomate, that means that it's going to have that coelom, one. It's also going to have all of the different tissue layers in contact with each other. So the mesoderm, the ectoderm, and the endoderm are all in some way connected and touching each other. And that allows for tissue communication. And when we have that coelom and we have that tissue communication, we can have organ formation and a really good amount of complexity in organ formation. So here's more of that coelom or that just that open space. All right. And so I'm not going to go into all of the different organs here because honestly, it's kind of hard to see when you can only see one piece of this at a time. But there's some really good diagrams that we provide you where you can see this entire cross section as one big whole piece. And then there are, look at the labels and see what each thing is pointing to. I think that actually the circle that we're looking at down here is one of the, one part of the circulatory system. So it's either the circulatory system or the nerve. And I think actually they might be like right on top of each other. But earthworms have a complete digestive system, which means that they have a mouth that goes through, that has a digestive system that goes all the way through the body, and then an anus. So it's complete. They luckily can eat on one end and excrete waste on the other end. They have a complete circulatory system and it's closed, which means that they have, just like we do, vessels that carry their blood from one spot in the body to the next. And they have a very sophisticated nervous system, meaning that they have nerves that run down the entire length of their body. And actually each different segment of an earthworm or a leech or whatever segmented worm we're looking at is going to have every single organ system that they need. So every single part, like, do they need a reproductive system? That's going to be in every single segment. Do they need the circulatory system? That's going to be in every single segment. So each of the different segments has all of the different organ systems that's necessary. I just want to come back to our earthworm one more time, just so that we can talk about, okay, so when we're looking at an earthworm, and I'll talk more about this when I do the dissection, but at the front, I know that this is his mouth because there's this organ here. See how it bulges out a little bit right there? That's the clitellum, and that's actually like a reproductive part of the earthworm, and we're not going to go into that for the purposes of this lab. But the reason I tell you that is you can always tell where the earthworm's mouth is going to be with where the clitellum is, because it's always skewed here. So if we looked at the entire length of this, and I could just zoom out and that would help us. I know that because that's there, this has to be the mouth end. And because this is all the way over here, it can't be that the mouth is here because it's too far away. So this is the tail end, okay? And just like us, imagine this was a human mouth. It's going to have stuff like a mouth, an esophagus, and then it's going to have intestine that run the entire length of it. And then back here, you're right, it's going to be the anus, all right? So very complex organ systems, very similar on some levels to a human. And it's hard for us to think about that because this is just our homely earthworm, right? But because of that coelom formation, because they are true coelomates, they really can have the complexity of organ formation and all of the rest of that goes along with coelom formation. All right, let's compare that really quickly to our roundworms. And I'm just going to show you one example of a roundworm because there's nothing to write home about. This is literally just kind of like a white sac here. I guess I could take it out. I'll do more of this when we do the dissection, but just so that you can see how simple this form is. So roundworms have a pseudocoelom, meaning that they have kind of a false coelom, and their body is pretty straightforward here. So this guy is probably old, so I'm not going to unbend him too much. But it's kind of just a white sac, a long white tube, and roundworms are generally parasitic, so they're pretty simple. There's not really much to write home on this specimen, but obviously it looks quite different than the segmented worms that we were looking at. And what I really want you to see is how much different it looks in the inside. So I'm going to do a cross section here of the earthworm, of the roundworm. So I want to make sure that I am. All right, and hopefully we can see this whole thing in the field of view. No, we cannot, because it's interesting, just so that you know, like what I'm seeing in my microscope is different than what you see. When I see in my microscope, I can see the entire circle or the circumference of this roundworm, but it just doesn't translate in the video. So I'm sorry about that. But what I can go down to the bottom here. And so here, what we have is the ectoderm or the epidermis, right? And so because this is a roundworm, the epidermis has a thick layer of cuticle. That's what it's going to shed when it undergoes ecdysis. And let's see if I can clean this slide up, y'all. All right, yeah, that's so much better. All right, and so we see the ectoderm and then we see the muscles. See those little fibers? Okay, so roundworms are pseudocoelomates. They have less complexity than a coelomate would or an earthworm. And so they only have one type of muscle. So instead of having longitudinal and circular, they only have longitudinal. And so their movements are going to be a lot more spastic, is the best way to put that, because they have less options on how they move. Whereas an earthworm with two different kinds of muscle groups, it can have really complex control over its muscles. It can have complex muscle movements. Yeah, it can do a lot more stuff, right? But we're looking at the roundworm. It's a little bit more simplistic. So let's move up here. And actually you can see there's a ton of space in this. So when we think about space, what we're really talking about is that coelom, right? And there's lots of little tiny, I want you to look at these balls. See how they look like little ornaments to me. There's lots of similar sized balls, and these are all organs. But interestingly, we talked about how coelom formation increases complexity for organ formation. In roundworms, you do have organ formation, but it's almost all centered on either the reproductive or the digestive system. So absolutely every one of these little balls that you're seeing, except for that really big one in the middle, is going to be associated with the reproductive system. And then this one in the middle, the kind of oblong one, that's going to be our digestive system. So this is a cross section. So this is a cross section of the intestines pretty much. And so what you see is that you have a digestive system, and then you have a bunch of reproductive organs. So pseudocoelomate means that there is a coelom, but that all three of the tissue layers are not necessarily in contact with each other. And the coelom is what prevents two of the layers from touching each other. So the endoderm, or the gastrovascular cavity, is surrounded by space, or that pseudocoelom, and that separates it completely from the mesoderm and the ectoderm. And so when you have that complete separation, what happens is that those tissue layers cannot effectively communicate with each other. And so what you find is that even though organ formation can happen because there is the space for it to happen, it doesn't necessarily happen in a very complex way because the tissue layers aren't communicating. And of course, complexity requires communication. All right, and the last group I want to look at, we're not going to actually look at any microscope slides of these. This is just going to be kind of a romp through the arthropods. So I just want to show you some arthropods. So here we have some, I think these are millipedes. And so we've got some millipedes. That's an arthropod. Here's a different, I don't like those samples that when the air's in the sample, when you see air bubbles, it makes it so that you can't see it as well. But here's a skeletonized version. Nope, it's not as good. Let's see. What I might have to do is just take them out of the jars, which is something that I don't normally do because it's kind of, they're in a solution that preserves them and it smells pretty funky. But I'll do it for you because otherwise you won't be able to see them. So we looked at some millipedes. Let's take a look at another arthropod. So let's take a look at this grasshopper. So we got a grasshopper and this scorpion thing. We've got millipede. Let's see if I can get this one out. Okay, I got it. And then we've got this shrimp. Okay, so the question is, what do all of these things have in common? Well, they all have this crunchy outer layer, which is the is what sheds in this. So these are surprisingly going to be related to our roundworm. And we talked about with the roundworm, we shed the cuticle or the outer layer, right? And in the insects, we shed the outer layer, but it's the harder outer layer, right? And so that's a characteristic, excuse me, of arthropods, right? And so this outer layer gets shed. Okay, so all of these have that crunchy outer layer, right? And so this is just, I want to show you how different these organisms might seem, but they have that characteristic or that critical innovation in common. And so that's why they are most closely related to each other in the animal kingdom. But all of these arthropods are coelomates, meaning that they have a true coelom formation, unlike our roundworm. So if you remember, our roundworm had what? Our roundworm had a pseudocoelom, but these arthropods have a coelom. And so what happens with the coelom is that you have complexity. So you'll see on your handout, we talk a lot about the different organ systems of arthropods, right? And so when we shed this, the exoskeleton of arthropods, right, they do that to grow. So as they get bigger and bigger, they're going to shed that exoskeleton and then grow a new exoskeleton so that they get bigger that way. The same thing is happening with this roundworm, but again, like very different version, right? So the roundworm is shedding that cuticle. So just good to compare these specimens, and I hope that that was really at least somewhat helpful to you in looking at these different specimens and how visually they look really different from the outside, but also when we're looking at cross sections of each of these different organisms. So we looked at the longitudinal section of a sponge. We looked at, we kind of looked at the hydra. It was because it was transparent, you could kind of see that it was the insides of it. We looked at a cross section of a flatworm or planarian. We looked at a cross section of the earthworm, and we looked at a cross section of the roundworm. And all of those were different both from the inside and from the outside. And so I just wanted to show you that, and hopefully when you watch this video, you're able to see those differences. So I really appreciate you joining me for this part. What I'm gonna do is I'm gonna add a second version where I'm gonna do a dissection of the roundworm and our earthworm. and the, let's see, roundworm versus earthworm. And so what we'll do is we'll do a side-by-side dissection of the earthworm and the roundworm, and we'll see how the organ systems look different, right? So we knew that the roundworm was a pseudocoelom and the earthworm is a coelom. And so we'll see what that looks like internally when we do those side-by-side dissections. But thank you so much for joining me. I really appreciate it. And I will follow up soon.